think they're both
All right,
Andy.
we're
All right,
being recorded.
I think we're sort of loaded up right now. So I've started recording the meeting. So anytime you guys want to start.
Okay.
Okay. Yeah, let's get started.
Yeah.
Thanks all the people out there who I just see your name on the side for joining us for our speaker series today. Our speaker is Jason Sylvan, who you can see on the screen. He's the one with the glasses. Jason got his PhD in marine biogeochemistry, biological oceanography from Rutgers University, his master's as well from there in 2008. He moved to LA right afterwards. He began a postdoc in deep sea geomicrobiology with Katrina Edwards, and he was a C-DEBI postdoc for 2 years as well. And about February 2013, right, he moved to the assistant professor research track within the Department of Biological Sciences at USC. And today he is going to talk to us about life in really old oceanic crust, some of his really cool new data, the quantifying cells there. And the way this is going to work is that if you have questions, you type them into the chat box at the bottom right of your screen. We're going to let Jason do his entire talk without interrupting him. And then he will field the questions at the end, directly interacting with him. So I'm going to turn off my webcam and hand it over to Jason.
Thank you, Julie. So bear with me for a sec while I switch to the actual presentation and then swap displays. So hopefully this is correct. Yeah, I'm going to spend the next 30 minutes or so talking to you about microbial life in old subsea floor basaltic crust. And I should probably start out by thanking you all for actually watching this over the World Cup, or perhaps that's on a side monitor, because that's going on at the same time. So after a brief introduction, I'm going to be talking to you about some work that I've been doing over the last few years, trying to develop methods for biomass quantification with samples of subsea floor basaltic crust, and then looking at microbial diversity and metagenomics from the same samples. And I'll finish up with a brief section about some culturing work that I've been doing. And so the kind of impetus for this work, or the motivation, can be summed up pretty succinctly in that a few sites and studies target the subsurface basaltic crust biosphere, yet we know this is a massive reservoir that water is moving through, and there's likely life all over the planet in this biosphere. So this figure here shows all of the sites that have been visited and sampled during the ocean drilling program and the integrated ocean drilling program, ODP in green and IODP in red. And so each circle indicates a site that was visited by either the JORDIS resolution or more recently the CHICU. And the sites that are slightly larger circles outlined in black are sites where microbiology studies were initiated. So you can see from this plot that the microbiology in drilling studies has been kind of a small proportion of the total work. And then on top of that, most of these sites are actually studies that were targeting the sedimentary biosphere. So there are a few sites that we do know quite a bit about, or at least a little bit. And so one of these would be the Atlantis Massif during IDP Expedition 304 and 305. Mason and all collected a bunch of samples and found evidence for hydrocarbon degraders up to 1,300 meters below seafloor, and also tried to do some cell biomass quantification, but found that things were either below detection or their methods just weren't quite working out for them. There has been a fair amount of work, as you can see from those papers, especially in recent years, looking at some sites off the coast of Washington on the Juan de Fuca Ridge, where using a series of quarks out there, scientists have found microbial nitrogen, sulfur, and carbon cycling, and also temporal variability in young ocean crust. And that work's resulted in a series of nice papers you can see listed there. And then I would also point out recently a paper came out from Lisa Negro looking at also a quark system where she found evidence for carbon fixation, looked at root viscogenes. However, you notice this is only 3 sites, and it's a very large ocean. So there's quite a bit to be discovered. And then the other thing that I would point out is that all of these sites that were previously studied are from young ocean crests, so less than 10 million years old. And so there's really a huge age gap in kind of the older ocean crests, and questions about whether or not there's anything there at all in terms of microbial life, and if so, what that is. So my work will be focusing on the site that's circled right here in red, or these sites, I should say. I do want to point out there was a little bit known, and so I published this figure in 2011 based on the few studies that were available at the time. But what we did here was looked at studies that had sequenced the 16S rRNA gene from bacteria, from subsea floor samples, also from studies that looked at hydrothermal plumes, deep seawater, hydrothermal fluids, and seafloor basalts. So basically environments where seawater is in contact with basaltic ocean crust. And the question we wanted to determine was whether or not the organisms in the subsurface basalt looked different from those communities in these other environments. And what this dendrogram is showing is that statistically that is the case. This was, should point out, based on a few studies at the time. However, initial work indicates that the microbes down there in subsurface crust are potentially unique. And again, we still don't know how many there are. So all of that kind of brought me to IODP Expedition 330, the Louisville Seamount Trail. This was a cruise that I joined as a microbiologist that went out from December 2010 to February 2011, during which 5 or 6 seamounts along the Louisville Seamount Trail were drilled by the JR resolution. I'm sorry, the JOIDES resolution. You can see here the sites that we sampled, how many samples I collected for microbiology from each site. This is New Zealand over here. So these sites are in the southwest Pacific Ocean, just northeast of New Zealand. The oldest site here, 1372, it's about 75 million years old, and is one of the last of the seamounts in this chain that's not yet subducted. The youngest site here is about 50 million years old. And this is a hotspot trail, and so these are extinct volcanoes that were formed in the same way that the Hawaiian Islands were formed. So one of the cool things about hotspot seamounts or volcanoes is that they grow through repeated volcanic eruptions. And so what you see here is the types of lithology that were recovered from site 1376, which was about 65 million years old. And you can see that as we go from the seafloor, about 0 meters below seafloor, down to about 180 meters below seafloor, these different colors just represent different lithologies that were collected. And then I have pictures of each of the samples that I collected and analyzed for microbiology. And so you could see both from looking at the different types of sandstone and carbonates that are bound stones and different types of basalts, that there's a lot of different lithologies in these types of systems. And so they're really interesting to study. And I like to say that multiple eruptions, it gives you different flow units and potentially isolated communities. And so if you're interested in questions about how substrate chemistry and mineralogy impacts microbial community structure, these are nice sites to visit because you can collect basically a lot of different types of samples from just one site. So I'm going to start off talking about the work that I did quantifying cells in these subsurface basaltic samples. I will start out with this figure published a few years ago by Jens Kalmar et al. A lot of people in the C-DEBI community have probably seen this, but this is the current synthesis of what we know about cell biomass in seafloor sediments. And one of the reasons I'm showing this is because we know pretty much nothing about cell biomass in ocean crust. And so we need something for which to compare any results that we get moving forward. And so what we have in this plot is kind of a classic on the x-axis here. We have log cell concentration of cells per cubic centimeter going from about 10 to the 2 cells per cubic centimeter up to 10 to the 10. And then on the y-axis, we have depth in meters below seafloor on a log scale. And so I'll just point out that the 0 is actually 1 meter below seafloor. And then we have 10 meters, 100 meters, and 1,000 meters below seafloor. And so in the black dots, we have kind of a summary of data from different ocean drilling program cruises, which largely visited continental shelf sites that had a little more biomass. And then in the different colors, we have data from different open ocean type sites. And so there are two patterns that I'd like to point out on this graph. One is that in general, when you go to a site and you collect some sediments, there's usually more cells closer to the seafloor and less cells as you go deeper down. And this, it doesn't matter so much how many cells you start with, the pattern remains the same. Even if you start with less cells, you still progressively get less as you go deeper. And then the other pattern is that it's actually quite heterogeneous. So depending on where you go, in black, like I said, we have sites that are closer to shore, versus in yellow, this is the South Pacific Gyre, where we have very low biomass and down to only 10 to the third or less cells per cubic centimeter. So after going on the cruise and collecting a bunch of samples and trying a few different ways to enumerate biomass here in the lab at USC, it was not going well. So I wrote a grant to C-DEBI and said, these collaborators in Japan, Yuki Morono and Fumio Nagaki, they have developed a really cool way to do a cell separation, and then it allows them to analyze really low biomass samples, mostly from sediments. And so I said it would be great if I could go over there and try it with my rock samples. And I got that grant, and so I went over there, and over the course of 2 years, I spent 6 weeks in their labs. They've been wonderful, gracious hosts. This is at JAMSTEC in Kochi. JAMSTEC is kind of like a national lab in Japan, and one of the reasons that their method is so successful is they have this really wonderful setup where you can see here, this is a clean room where all they do is cell counts, and you wear a mask and gloves and jackets, and it's the air is filtered, and so it allows you to get really low detection limits. And so working with Yuki and Fumio, we adapted Yuki's recently published method. So this was published last year in Environmental Microbiology for working with sediment samples. But the basic idea is that you take a slurry of your sample. So I started out in this case with frozen rock samples that were collected from cores. I always made sure during the cruise to collect samples, basically split the core and collect from the inside, because previous work, especially by Mark Lever, has shown that the outside of cores, and especially rock cores, can be contaminated. So you collect samples just from the inside, and then froze that, and then back in the lab at JAMSTEC, I took those samples, crushed them in a mortar and pestle, and preserved them. And then first, you shake it quite a bit, and then you use a sonicator, and both of those are in efforts to basically knock the cells off of the rocks. And then you take this slurry that you have and add it to, this is a 15 mil centrifuge tube, which has a gradient of nicodens. And the idea here is that nicodens is more dense than cells. And so if you centrifuge this, the cells should not pass through the nicodens. And at the end, you should have your rocks at the bottom of the tube and your cells suspended somewhere in one of these layers. The paper that came out last year showed that using these multiple layers from nicodens as well as sodium polytungstate actually gives you more efficient recovery than using just a single density layer of nicodens. And so you can see over here in this picture 2 samples that were actually spun, and indeed this method seems to work. And so you see down here at the bottom, this is fragments of rocks, and so you do not collect that, but then you collect everything that's above it, including some of these very, very fine particles from your samples, and filter them and stain it with cyber green, and then go to a microscope. And I would say typically I might count somewhere between 600 and 800 fields of view for any one sample, and a lot of times I see something that looks like this, which is not much going on. But then every now and then you see something that looks like this. And so this is a happy microbial cell from subsurface basaltic crust. And we get really excited, and we do that for a bunch of samples, and we get something that looks like this. And so what you see here is a summary of my work from 3 different sites. So this is site 1374. This has samples down to about 500 meters below seafloor. sorry about that, site 1376 and site 1372. These 3 sites are all from the Louisville Seamount cruise. And then also I have some samples that were provided by Fumio from site 1301B. And this is the site where, on the Juan de Fuca, that some of the work in Mark Lieber's paper, some of those samples came from that site. And so I want to walk you through these results. There are basically 2 big things that I see here that have me really excited. So I have the sedimentary plot up for reference. And so the first thing that you'll notice, there's this dotted line. Basically, anything to the left of the dotted line is below our detection limit. We through a series of blanks, figured out that about 10 to the second cells is what we feel comfortable calling accurate and real. So anything to the right of that, we believe. This one sample is from South Pacific gyre sediment, basically at the top of the seamounts. These seamounts kind of cut into the South Pacific gyre. And this number matches up pretty well at, not quite about 7 or 8 meters below seafloor. We have a little over 10 to the third cells per cubic centimeter, which you see is would be right here. So that matches up pretty well to what previous samples have shown. And so if you look at the rock samples, we see that most of the biomass goes from either below detection up to about 10 to the 4th cells per cubic centimeter. If we circle just that area of the plot, so this is where all of my data lies that is above detection limit, and put that same data range on the sedimentary plot, what we see is that biomass is quite low, and it's in the lowest corner of what we've seen in sedimentary systems before. So this tells us that there's the density of cells down there, at least at these sites, is not great. But the flip side of that is that the volume of the substrate is great. And so it is still likely that there's quite a lot of biomass in these systems. And then the other thing that I want to point out is in sedimentary systems, as I mentioned earlier, you see these kind of, logarithmically linear decreases of biomass with depth. If you look at just one site for the basaltic systems, you see, if you look at just the blue circles, you can see that it kind of jumps around quite a bit. And that's true for all the sites. So we do not see this linear decrease with depth in the amount of biomass, and therefore that indicates to me that there is something fundamentally different about life in subsea floor ocean crust than there is in life in subsea floor sediments. So I think that's really exciting. From a lot of these same samples, subsamples have been sent off to Jeff Alt at University of Michigan, and we're hoping to get some data on organic carbon and delta carbon 13 from the same samples. So stay tuned, because I'm really excited about this data, and this is stuff that I hope to write up and get out soon. So despite those really low biomass numbers, I was able to get DNA from a subset of those samples. So I'm going to talk to you now a little bit about that work and looking at microbial diversity in some of these samples. So going back to the map of where these sites are, I focused here on 2 of the sites, site 1374, which is this site. The reason this is a lot smaller than this is because they're to scale. And so at site 1374, I collected samples from 40 meters below seafloor down to 490 meters below seafloor. At site 1376, I have samples from 30 meters below seafloor down to about 180 or so meters below seafloor. So there's 15 samples using about 2 to 3 grams of rock for each sample. I did a DNA extraction. And as part of the census of deep life, I had these sequenced for the 16S rRNA gene using 454 sequencing of the V4, V6 region. So this returns about 450 base pair long sequences. So when you do that, these are the results. I'm going to kind of walk you through this. It's a lot to look at. The one thing I want to point out in the beginning is there's 15 samples here. For all of these, I also sampled drill fluid from the cruise and a blank of the kind of cleanup kit that I used during the extraction. And so any OTUs, operational taxonomic units, that were detected in those samples were previously removed from the results that you see here. And so these numbers, depending on the sample, range from just one sample that had about 700 or 800 sequences up to about 10,000 sequences per sample. And so What you see here is each bar here is a different sample looking at the bacterial classes that were detected in these samples. In red, we have all of the sites from 1374 going from shallowest, again, at about 40 meters below seafloor to about 500 meters below seafloor at 7468. And site 1376 going from about 30 meters below seafloor down to about 180 meters below seafloor at site sample 7623. I like to look at these types of plots just to see if there are obvious patterns that really pop out. I would say in this case, I don't know that's the case. And so I'll move on to just who are kind of the major groups that are there, and that's something a little more obvious. And so there's kind of 8 bacterial classes that seem to be the most abundant in all the samples, and these include Actinobacteria, Flavobacteria, and Sphingobacteria in the phylum Bacteroidetes, the Bacilli and Clostridia in the phylum Firmicutes, and alpha, beta, and gamma proteobacteria. If you compare statistically how similar these different communities are, a hypothesis going into that might be that samples from the same seamount would cluster together. This is using Bray-Curtis analysis, which accounts for abundance and presence or absence of different operational taxonomic units. But looking at the results of this plot, I don't necessarily see any obvious patterns. I also looked at this using just presence or absence of organisms, wondering if maybe that would provide a clearer question. And the answer was actually no. So it's not like all the samples from one seamount kind of fall out next to each other, or there's some pattern with the deepest samples kind of next to each other and the shallowest next to each other. And I think that part of that is if you look back at the samples that were actually collected, right, they were quite far apart from each other. And so if you look at just, this sample was collected 40 meters below seafloor, and this is 75 meters below seafloor, and I was specifically looking to collect different types of lithologies. And so I think we're just seeing a lot of heterogeneity in a system that itself is lithologically heterogeneous. However, if you kind of step back and instead of looking at individual samples, just go more broadly and looking at, well, what types of organisms in general are in this environment, you could put together a tree that looks like this. And so what I was basically looking for here is organisms that were present in more than one sample. I'm sorry, 3 or more samples, I should say, or if they were present at, a lot in one sample, like more than 1% of a single sample. And so this is the result of looking for those different, this is basically genera. And what we notice is if you can assign putative functions to some of these genera based on what we know from cultured isolates, we see a lot of organisms that are involved in sulfur, nitrogen, and metal redox processes, as well as hydrocarbon oxidation. Especially here in the beta proteobacteria, we see a lot of genera that are involved in hydrocarbon oxidation, and that seems to support what was seen, especially at the Atlantis Massif sample study. And then also a lot of organisms involved in kind of sulfur cycling, like we saw on the Juan de Fuca ridge. So I think that's pretty exciting. We can also try to look for not only just what individual types of organisms might be doing, but if there are associations between groups of organisms. And so this is called network analysis, and Luke talked about this before in his talk last month. But the basic idea here is that if you take all of the operational taxonomic units that you detect in all of your samples and also add environmental variables, and so in this case, since these samples were collected during IODP cruise, I have, for specific lithologies, I have data about elemental composition and porosity, and I also have my biomass data. And you make a matrix of correlations between all of these variables. You can kind of visualize that matrix and try to tease apart if there are patterns between different organisms or between organisms and different environmental variables. And so I show up here first kind of a simpler version of this. This comes from a paper by Josh Steele et al. In Jed Furman's lab here at USC. This is one of the first papers to do this, and they were looking at surface water column communities off the coast of California. And in this case, we have a particular eukaryote, this is a stromenopile, actually, the diamond. So all the diamonds in this figure are different eukaryotic species. All of the circles are different bacterial species, and you could see that these arrows indicate these species are all somehow correlated with each other, the presence or absence and abundance of these species. And then additionally, an environmental variable, in this case chlorophyll A, also seems to impact the presence and abundance of these species. So if you do that with my data, you get something that at the very kind of broadest scale looks like this. And so what we have here is all the OTUs and environmental variables. Environmental variables, in this case, are in black diamonds. You see that for the most part, a lot of them kind of fall out on their own down here in the corner, but there are a few that seem to be related to at least a few different OTUs. And then the other thing that we notice is if I basically color-coded each OTU according to which sample they were most abundant. And what that results in is basically these kind of clouds of single samples. And so here you see all of these OTUs were most abundant in sample 7452. And if you go down here, all of these OTUs were most abundant in sample 7414. And there are a few cases like in this area where you see a little more mixing going on. So that's something of interest to figure out what those are moving forward. But I do want to point out this may not necessarily be that surprising, given that if you look, the Bray-Curtis analysis indicated that there was a lot of heterogeneity, and if you looked at just the taxonomic distributions, there weren't any obvious patterns. And so I think that that is possibly what's causing the patterns that you see here. However, we can zoom in on these features and see what the actual organisms are that are in these clouds. And so I'm going to zoom in on this one feature here, which is from sample 7604, which was the shallowest sample at the second site, so about 30 meters below seafloor. And what you see, this is all of these organisms, because they're all solid lines, that indicates they're all positively correlated to each other. So basically, the presence of all of these organisms seems to be correlated with each other on some level. And then the size of the circle is an indication of how abundant in the whole study that these particular organisms were. And so here you can see there's Nitrosomonaceae, which is a nitrite oxidizing member of the beta proteobacteria that seems to be correlated with a lot of organisms that, if you look them up, are commonly found in soil environments or other kind of subsurface environments. And so I think I need to do a little more thinking about this, but it looks like it's definitely informative and indicative, at least, of organisms that seem to be kind of co-present in samples and that may possibly be meaningful. So I'd like to kind of move on beyond just looking at 16S RNA and kind of doing some hand-waving about what might be happening. And to do that, I'm looking at metagenomes from just 3 of these samples. So I selected 3 samples from site 1376, and through a second round of Census of Deep Life funding, had these the metagenome sequence. For these samples, I actually had to start with quite a bit more material to get enough DNA for a metagenome. And so I started with 25 grams of sample. And for the 3 different samples, that resulted in about 10 to 100 total nanograms of DNA. So for people doing this kind of work, hopefully that gives you a reference point, helps you out for how much sample you really need to start with. Due to some issues with potential contamination, we did not use the same cleanup procedures for these samples. And so they should be a little cleaner. And then we sent them to the Marine Biological Laboratory, and Sharon Grimm in Hilary Marston's lab sequenced the metagenomes on a high-seq using paired end under 50 base pair reads. And so what I'm going to talk about for the next few slides is this kind of initial analysis and kind of a pipeline that I'm developing with Tada Ben-Tully, who is now a C-DEBI kind of bioinformatics postdoc. And part of Ben's role is to help the C-DEBI community. So people that have omics type data but aren't exactly sure what they might want to do with it can go to Ben. And so part of this is advertising my data, but part of this is actually also advertising Ben services. And so if there are people out there listening to this that have data and they're not sure what to do with it, I urge you to send email to Ben, and you could figure out a path forward to try to get something out of your data. So in our case, and this seems to be pretty helpful for us, the first thing we did was use a program called meta-RNA and basically go through all of the metagenome and fish out the pieces that were just for 16S rRNA, and then use a second program. So here, these different colors indicate different short fragments of 16S rRNA from different organisms, and then used a different program called eMERGE to basically take those pieces, compare them to a database, and put them together. And identify who's actually there using a Sylvan database. And so what this tells you is basically the types of organisms that are in your metagenome, and so you have some expectation for how clean that metagenome is and what to expect from the assembly when you start looking at functional genes. One of the things that this told us is that one of our samples, unfortunately, was not terribly clean. So moving forward, we're kind of focusing on 2 samples and decided to do joint assembly of those samples. So one of the cool things from eMERGE is it gives you some 16S data, and so it lets you, kind of figure out what organisms you might expect to find in the larger metagenome. And so I made a phylogenetic tree of just those 16S here, and just have labeled all of these branches represent 16S RNA sequences from the metagenome that were assembled, but I just kind of put a few out there that I personally think are interesting, things like nitrosamonidaceae, which I mentioned earlier in the network analysis, as well as Ralstonia and a bunch of Bacteroidetes, and it would be really great to find some of these in the metagenome and assign some sort of function to them. So taking the metagenome, we used a program called IDBAUD to assemble it, and then took all contigs that were greater than 2,500 base pairs and clustered and mapped them using a program, Emerging Self-Organizing Maps, or ESOMs. People may have heard of this, which uses tetranucleotide frequencies, percent GC, depth of coverage, and the percent usage of a select set of amino acids to kind of group sequences together, hopefully in different bins that are representative of one genome, or kind of a pan-genome of one species or genus. And so this is what that looks like initially, but you can then use the program to make these definitions between different genomes a little more obvious by coloring them in. And so I do want to point out it's a little bit tiled, so that it goes off the top and kind of starts repeating on the bottom. But what you see here is that from the initial assembly, that we have clearly 5, so dark blue 1, green, yellow, pink, and this kind of light blue. These are 5 different genomic bins that are well defined. This is pretty new, so we're not sure exactly what they are yet, but over the next week we'll be able to figure that out and decide moving forward, are these things that we really want to focus on, and what kind of story do we want to tell with this data. And then over here, kind of in this area, what you're seeing is a lot of sequences that are unresolved. And so this could all be from one group of organisms, or it could just be a mishmash of sequences that just are not resolved for whatever reason. So pretty excited about that. So, and then finally, I just wanted to kind of advertise, I'm really excited. So beyond doing cell biomass quantification and also looking at microbial diversity, I started a series of enrichments during the cruise, and has had some success with that. So this is one of those stories. This is Bacillus rigili profundus, which was a new species of bacillus that was most closely related to bacillus from serpentinizing environments and some subsurface terrestrial environments. So I thought that was pretty exciting, and moved forward with like kind of a official description of this species. And one of the fun things about it is that as a spore, it sporulates, and as a spore, here you can see pictures of the spores taken by Lily Mompert here at USC. As a spore, it actually oxidizes manganese, which is pretty neat. And so we'll see if the name flies, but I called it rigili profundus. That should be you, sorry. Because it was sampled from Rigel Geo and deep within it. So deep within Rigel. Hopefully the people that do all the official naming things are okay with that. And so to summarize, What I found from these samples, kind of looking at old ocean crust, 65, 75 million year old crust, is that there's low biomass from below detection to up to about 10 to the fourth cells per cubic centimeter of rock, which is as low as nearby sediments in the South Pacific Gyre. We found bacterial populations related to sulfur, nitrogen, and metal redox cycling and oxidation of hydrocarbons. We found correlated bacterial taxa that were uncovered through network analysis. Initial metagenomic analysis is promising, and future work will provide evidence for putative microbial functions. And finally, novel isolates, as Bacillus was one, there are some others, allow for future testing of hypotheses in a controlled laboratory environment. I'd like to just kind of finish up with my acknowledgments, especially Katrina Edwards and her lab. I was a postdoc during some of this work, and now I still continue to use her lab for my work. Jan Ament as well for his assistance and the use of his lab, and a whole series of people that helped on the cruise with the sequencing, especially Sharon Grimm and Hillary Morrison. Finally, Anthony Koppers, Toshi Yamazaki, these were the chief scientists on Expedition 330, and Jorg Geldmacher was the staff scientist. And without them inviting me to join the cruise and their assistance during the cruise, none of this would have been possible. So with that, I will finish up and say thank you. So that I can see the questions, I'm going to close the slideshow, but I'll keep it open in case I need to bring up any slides. So thank you for listening, and I'll take any questions. Oh, start my webcam. Okay, so the first question is from Karen Lloyd, and the question was, did you see any archaea? So the initial answer to that will be no, and I'll say two things. So one is that from the DNA extractions, I did also do qPCR for 16S RNA, and because there were issues with this subtractive type analysis, the numbers for the bacteria, I think, are a little bit suspect, and so they're not something that I would rely on. However, archaea were not amplifiable, at least, and so I think that that does indicate that in this particular environment, for whatever reason, they don't seem to be abundant. A better answer to that will come in the metagenome, and if we see indications of bacterial sequences there. However, in these kind of initial analysis of the 16S fragments that we could pull out, none of them belong to archaea either, and so initially it's looking like they're not very abundant in this particular environment. Luke McKay asks,
Yes,
do I think, oh, yeah, I'm sorry, Matt.
Thanks. No problem.
Oh, I'm sorry, it starts sharing. I can see myself on my own screen, which is really helpful, but is that better?
Perfect, thanks.
Okay. Sure, sorry about that. Luke McKay says, do I think the gamma proteobacteria can imply some subsurface source of oxygen? This is a really good question. So going into this, I knew that one of the great things about the cork studies is that you, because you are going back and able to kind of sample again, you know a lot more about environmental context with these samples because they're just cores, and they were not corked afterwards, and we don't have any pore waters. We kind of have no idea. I would say I agree with Luke. I think that I see a lot of organisms that are either aerobic or facultatively anaerobic, and it indicates that there is some subsurface source of oxygen. Because these are seamounts, it is possible that water has moved through them more recently than maybe if they were, below the kind of seafloor. It had never risen up at some point. So I would say the answer is we can't say for sure, but I do think that it implies that oxygen may be present in this environment. Steve Daunt asks, or the Daunt Lab, did the amount of DNA you get out of the 25 grams of rock jive with the number of cells you counted, and would you expect in that much rock? That's clarified that that's actually John asking. I think loosely, yes, actually. So the one sample, there were 3 metagenomes in the one sample that I said kind of we were able to sequence, but it ended up being mostly organisms indicative of some sort of contamination. I went back to the cell counts, which unfortunately were done after some of the molecular work, just because of the way things went. And it turned out that was one of the samples that had below detection cells, and so I think that on some level that is true. Oh, I'm sorry. Just one of the questions went past me without me noticing. I should go higher up. But so I see Brandy Reese was wondering, can you repeat what program you used to assemble the metagenome? Yes, I used a program called IDBA-UD. It uses iterative k-mers. It basically goes from small k-mers to larger k-mers and uses the contigs from the previous round as the initial sequences for the next round. And it seems to be pretty popular, but there are other ones out there. And I would say, again, that if people have that kind of data and they're not sure where to start, which I can relate to, definitely to send them an email. But yeah, we used IDBA-UD. Mike, I assume Mike Lee asks, I think he mentioned, oh, let me see, I'm sorry. Why did I use different percentage of nicodens, tuba S? That's a good question. So older versions of that method with the cell separation had used just a single layer of nicodens. And that can work well depending on the type of samples, but it was noticed, especially with sediments, that some of the cells were kind of sticking to the particles and being sedimented out after the centrifugation, and that by using the different layers of nicodens, the percent recovery was much higher. And this was basically taking sediment, adding E. coli to it, so you know what you start with, and then doing experiments. And they found that having the multiple layers seems to help. I don't know if it's that it catches some of the smaller particles in suspension, or if as the cells attached to particles are moving through successively more dense layers, they get knocked off. But either way, the result is that the percent recovery is high. That's why there's multiple layers. So Luke asks, in the network analysis, what was the difference between two OTUs close together that were connected with the line and another two that were close together but not connected? So I used Cytoscape to make that figure, and the length between two OTUs is actually not informative. What was informative is the thickness of the line and whether it was solid or dotted. So dotted meant a negative correlation. So two things that were kind of next to each other but not connected means that there actually was not a correlation between those. It's a lot to look at kind of all at once, but when you kind of zoom in on the different kind of clusters, it becomes a little more obvious. I did notice, I liked Luke's network. It was like super 3D and exciting looking. Oh, a few people are typing, so I will wait patiently. Tina asks, hello Tina, what was the culture media that you have had success in enriching the new species? Oh, that's a good question. So that the bacillus that I isolated, and also some Halomonas that were not new species, but so just different isolates from what's been grown before. I grew with dilute marine broth. So when I got out to the ship, I had brought media that targeted both heterotrophic and autotrophic sulfur oxidizers, iron oxidizers, iron reducers, sulfate reducers, and methanogens. But when I got out to the ship, from the previous cruise was 329, the South Pacific Gyre, there was a lot of media. One of them was marine broth, And I know that that's basically like chocolate candy for microbes, so what I did was dilute it quite a bit. And so the bacillus that was grown, the initial enrichment media was marine broth diluted 100-fold, so basically 1% marine broth. And that just targets random heterotrophs, I would say. Roslyn Plus Ben asks, from the results you saw for 16S amplicons, looking at the Bray-Curtis analysis, would you expect anything other than positive correlation between OTUs from the same sample? That's a really good question. I would say looking at the Bray-Curtis thing, I would say I would hope that there might be some OTUs. I mean, that, I guess I'm not surprised that the overall pattern indicates that there's mostly correlation between OTUs from the same sample. What I was hoping for or interested in was seeing if there were, specific OTUs, even if just a few, that kind of were in multiple samples that seem to be correlated with each other. And I think I need to still look at that a little deeper, but the pattern seems to mimic what we saw in the Bray-Curtis analysis. There's still a few people typing. Randy said, you mentioned several metabolism genes in your metagenome. Did you notice any functional genes for cell physiology that you did not expect? So we have in the metagenome, currently we've only been to kind of put together those 16S RNA genes, and I'm hoping for metabolism genes. I assume that there will be at least some, especially in those genomic bins, but I haven't analyzed that yet, so I don't have an answer to that yet. But I hope to sometime over the summer. And then Tina Lynn asks, I'm curious about how the new isolate is oxidizing manganese, and that was why the culture medium. Oh, okay. So actually, the reason that I tested for manganese oxidation, if you look at work out of Brad Tebow's lab and Ken Nielsen as well, it's just known that in spore coats there are genes for oxidation of manganese. And so I thought, since I have a bacillus and it's from a subsurface environment and some of the other organisms from surface environments, especially terrestrial ones, there was a paper by Lisa Mayhew that she had a bunch of bacillus that were isolated from terrestrial subsurface sites that could all oxidize manganese. I thought I would do that test. And so that's the original enrichment media was not something targeting manganese oxidation, but that was something that came up later. Sean Youngbooth asked, what is the downhole temperature of the collected rocks, and does that match the psychrophilic, mesophilic, hemophilic signature of microbial communities detected? So downhole temperature was estimated to never be above 10 degrees, and that's because these are old extinct seamounts, and even at 500 meters below, I think that was the one that was maybe 10 degrees. And so I would say the answer to the question is mostly yes. I didn't really detect that there were a few organisms that might be thermophilic, but on the whole, I seem to detect more psychrophilic and mesophilic organisms, and so I think that does match with what we were expecting. Cool. Quick follow-up from Rosalind and Ben. To see negative correlations, would that require looking at long-range differences? So always comparing at least two samples. I did actually wonder, so those of you not at USC did not see Jacob Cram, one of Ted Furman's students, give a really nice defense yesterday where he used a similar type of analysis and saw really cool patterns in time series over 10 years. And so they have a lot of samples, and it's over a long time. And I was wondering if perhaps more samples or just temporal change would allow me to see negative correlations. I thought maybe with all the different types of lithology sample that you might be able to see something, but that just wasn't the case. Thanks. Well, I think the questions have slowed down, so unless any more pop up, thank you for listening, and thanks, Matt, for your help, and Julie for introducing me.
Thanks, Jason.
Thanks, Dave.
And for everyone's reference, this will be posted online in a few days for anyone who missed it. And stay tuned. I think the next talk is Julie Meyer in August.
Yay. Bye.